to introduce our speaker today, Dr. Enkeleida Lushi, which is called Kala. Kala was actually here a few years ago as a postdoc with our colleague Patio, what I can't pronounce her last name, but many of us know, who sort of at that time just moved to Northwestern University, but Kala was on campus doing their postdoctoral work. So we had many friendly conversations. We never collaborated at that time, but years passed now, we're starting to collaborate. Kala did for undergraduate and master study at Simon Fraser University in Canada. And then did her PhD at New York University at Current Institute at that time also, I think, with Michael Shelley. She then did a couple of postdocs, one at the University of Cambridge with Professor Ray Goldstein, and then, as I said, and then briefly with Patio before launching on their faculty position in 2018 at New Jersey Institute of Technology. Sometimes I feel like a mixed feeling if I should mention all the names somebody has worked with, but in reality, I think it's just a way for, especially for older scientists, it's just for them to anchor acquaintances, So it's just all to help people project what they were working on. For Kala's work in particular, Trent as an applied mathematician has done a lot of computer simulations, but with really in-depth knowledge of hydrodynamic interaction. That's the hallmark of Michael Shelley's work, and then also her work for postdoc. And then a few months ago, I think when we were looking into swarming dynamics, and then I found Kala's 2014 paper. At that time, it has 580 citations, probably more now. And so this is the work we are very interested in. So we will just hear from Kala directly.
It's great to be back here, and I'll tell you about micro-swimmers in confinement, and also kind of how I've arrived to this topic and what interest, how I learned about this, and what kind of defined this trajectory of all these various visits and postdocs. So my background is in math and simulations, right? So along the way, I got to learn about interesting problems, and a problem that is of interest is collective motion. So, and this can be seen in nature at all scales, from bacteria, even smaller units, to spermatozoa, now artificial colloids, birds, fishes. You see this, even humans, sheep, and so on. And the big questions are, well, how do you get collective motion? How does this arise? So you have, in most of the cases here, self-propelled units. One of them moves forward and then interacts with the others. And these interactions will depend on the medium, on the species, right? They may not always be mechanical, they vary. And then you get collective behavior. So micro-swimmers, like a physical realization of this living active matter. So active matter is the field that studies collective motion. There's a lot of interest, right, to kind of figure out how we go from this to this, right? Figuring out what are these interactions, can we understand, right, how these arise. You can imagine from different scales and mediums, this can vary a lot. And people study this, people with different backgrounds, from experiments to theory to simulations. So I'll tell kind of my own story. The aims of kind of what I try to do with the group is math modeling of the swimmers, in particular building the models for the specific type of swimmers and include the correct physics, fluids, there's anything in the environment, there's any chemistry, include the particulars of the swimmers. Explain later why that's important. And very importantly, keep the model as simple as you can get, but not simpler. Right? And once these models are built, then there is a lot of work on the algorithm development, right? Doing simulations of this. So once you trust the model, and you can simulate many of these, and then get into all of this, how to make this efficiently, include all of these interactions in an efficient way, many units interacting, and implement them, and ideally make the method versatile, so you can tweak it and apply it to other situations. And last but not least, this is a very important point, is validate the simulations and the models with experiments. Go back and forth, make sure that what one is capturing is something realistic. And then once you kind of converge on a model that you can trust, you can use them to study phenomena. So all of these are intertwined. So it's not doing one or the other, right? It's kind of doing all of this combined. So I kind of focus on the microswimmer scale, but not only. And there the question is microswimmers. Right? So things can have natural microswimmers like bacteria, swimming bacteria. They're only at that stage, particular stage of life. Things like Enkeleida, Lushi. And if you're looking at swimmers, ideally you want to categorize them, right? Or make modeling easier. And for these, right, swim bacteria, they have the flagella, superflagellum, a little flagella in the back. And they, so the flagella are in the rear. So the cell self-propels, and as it moves, it disturbs the fluid flow. If you look at this fluid flow, roughly you get this kind of hyperbolic fluid flow, with the streamlines coming in the middle of the body and going out the front and back, right? And it's called the pusher microswim because it pushes itself forward with the flagellum. Now this is only the far-field fluid flow, right? Near the body, of course, you'll have more variations and everything, but it's still useful because you can get this, right, with a dipole, right, two singularities that are opposing. The other studied microswimmer, this other model microswimmer, is micro-algae. It's roughly the opposite. It has just two flagella in front. It does something like a breaststroke. And it's called the puller, prototypical puller, because it pulls itself forward, right, using the flagella. And interestingly, if you average the flow field, right, the disturbance fluid flow that it generates over the cycle, it's roughly, sign-wise, the opposite that of the bacteria. Right? So the fluid flow is coming in on the front and back and leaving at the sides. It's a bit more complicated than that, but right, to that leading kind of order, that's the fluid flow. Spermatozoa are another type of swimmer. They're slightly bigger. Right, so the bacteria, you're looking at a few microns for the body, spermatozoa, maybe 50 microns. Different mode of locomotion, they just have one flagellum that they undulate, that's how they push themselves forward, but it's also pusher microswimmers. So if you average the fluid flow, right, over one undulation, you get this pusher flow. Now, that's useful if you're doing math, because it's a sign change, right? So if you want to include these fluid disturbance flows, that's kind of the starting point. And then I'll mention in the end these sporadic swimmers. So these are Janus particles, a few microns long, and if you put them in hydrogen peroxide, they swim. And the majority of the studies focus on spheres or rods, those are easier to make, but we can make them pushers or pullers or more. And study hydrodynamics. So this has been studied quite a lot, I guess, in the last more than 2 decades, but there was a lot of interest, right, in how you get this so-called bacterial turbulence. Quotation marks, because it's not high enough number fluid flow. You just have a lot of bacteria, elongated bacillus subtilis, 4 or 5 micron longs, right, and put together in a drop, right, they start to collectively move together, right, in this quasi-chaotic fashion. So there was a lot of questions. How is this collective motion emerging? Remember, they are not thinking, right? They don't have a leader to tell them where to go. So anything that arises is mechanics, right? Or chemistry, maybe if there is something. So there's been a lot of simulations, of course, a lot of continuum theories. So the most prevalent model, and we'll talk it, is the squirmers, so modeling them as spheres. There have been simulations with just rods, but not necessarily taking into account lesions, and mine are here in between. So ellipsoid, but with hydrodynamics. So there's been a lot of work on this, and what's been known for a while is that if you get two pushers, in this case bacteria, they co-swim a little bit, right? Because of the shape, they're elongated, but also because these fluid flows, the disturbance fluid flows, push them together and keep them together. So for pushers, co-swimming is more likely. And this is the assumption of many continuum theories or V-check-like models. And we'll come back to this later. So where do we start? We want to make models of microswimmers, right? And there are models of various complexity, dumbbells, ellipsoids, various like dipoles. This is the squirmer, by far the most prevalent, so a sphere with varying tangential velocity. With some effort, one can make that a different shape, right? But it's still difficult. So what do we want to keep in mind, right? Well, swimmers live in fluid, not in vacuum. It seems like a silly point to make, but they are. And if they swim, they disturb the surrounding fluid flow. Hence, they disturb neighbors or swimmers that are nearby. They do collide with neighbors and surfaces, and that becomes important, right? Like the shape becomes important there, how that collision happens. And most are not spheres. So if you try to simplify to the sphere, it's a bit too much. So if you're thinking as a modeler, you want to make things as simple as you can, but not simpler. Right? So imagine you're Picasso and you have to make a big painting of 10,000 bulls. It will be difficult to draw everything in detail. So you want to distill this as much as you can to something that gives the essentials. You can still understand it's a bull, right? It has legs, a tail, horns, right? So you get the essentials of what animal this is, and it's easier to draw, right? But don't want to go too far and make a spherical bull because maybe that's too simplified, right? So as a modeler, you want to get to something that is very simple, right? Simple model, because that will make it easier to simulate, right? If you're simulating many. So a model that I started out with is just tracing the center of mass and the direction of movement, and having this as ellipsoids, self-propelling ellipsoids. And for each swimmer, just tracing center of mass and the direction. So this is what these equations show. They're complicated, but it's saying a swimmer self-propels. If there is any other flow around, it gets affected. There is forces and torques, so it collides with neighbors. And this is just rotation and fluid flow, background fluid flow. One thing we added up these fluid flow disturbances. Right? And so each swimmer is an ellipsoid, and then you have this force dipole for the bacteria. So it doesn't seem complicated, and it's not if you write the model, right? So you have Stokes equations with adding the action of all these forces. And it is a couple of dynamics, right, of the swimmers that are anywhere in the domain, and you have to then solve these hydrodynamic interactions. You're mostly familiar, suppose, with Stokes singularities and adding, right, those. Here we're doing something different, right? We're coupling these forces to the fluid flow, which is solved on a grid. Right? So this is what these immersed boundaries methods for fluid structure interactions, this is what they do. They couple the forces and effects of the body and the fluid to the mesh, right? In which you solve for the fluid flow. So, and obviously you want to solve for the fluid flow using whatever easy method there is, like FFTs, and this, but we include the collisions, right, the shape of the swimmer, not just in the fluid effect, but also in how it collides with others. So forces and torques if they collide. And it's fast numerically. And most importantly, the method is versatile. You can include a variety of boundaries. And the particles, of course, don't need to be uniform, speed or shape or type, right, which you can easily do with continuum theories. And the fluid flow doesn't have to be Newtonian, or for that matter, Stokes flow. You can do viscoelastic flows. So we get collective dynamics, right? And here is like a 2D simulation. And here it's 3D, elongated, moving fast. And So, okay, this is good, right? In computational methods, you show this to people, that so many people have simulations, right? So, what's the challenge? The challenge is confinement. You look at swimmers in nature, in the lab, they're always in some confinement. So, here I'm showing some examples. Micro-algae in a device that kind of sorts them. Here is a bacteria going in an undulating channel, and because of the interactions with the surfaces, they can start forming biofilms. Obviously, bacteria sticking on the surface. Here is spermatozoa in a microchannel that has an asymmetry, which induces them to start moving in a direction. And even your prototypical swimmers, like bacteria, like it lives in soil, tissues, all sorts of things. Microalgae are used in photobioreactors, which are porous kind of structures. Spermatozoa, they have to navigate a network of filaments, right, in liquids. So always in some confinement. So the questions are, okay, we want to study this. They obviously live in confinement and have evolved in confinement. So I happened to be in Cambridge when this paper was being published, and kind of started curiosity on it. So the group of Ray Goldstein and PhD student Hugo, they confined the bacteria in a drop and between two cover slips, so you get this kind of cylindrical shaped drop. And then by themselves they organize. You see this, the drops in the same size. They start to organize with bacteria in the center or in the bulk moving in one direction. But then you also see a boundary layer, right, that is moving in the opposite direction. And the suspension is stable for as long as they have oxygen. And the question was, well, what is causing this? What can we do? So I modified the numerical method to simulate this water-oil drop interface, which is very basic simulation. So, and it's that same method, right? So swimmers collide, there is hydrodynamics, and also interaction with the boundary. So what happens? You have self-propelling units. They end up at the interface, and then you can see the hydrodynamics is pushing them against the surface. It's kind of helping to keep them to the surface, and they're swimming alongside the surface at a very, very low angle. And you don't see any collective motion, but bacteria like surfaces interfaces, right? You see that in experiments. Now, what happens if you have a few more? They end up at the boundary, right? But then they have to fight the others because there's quite a few. And then it becomes musical chairs for bacteria, right? So they start to push others out of the way. And remember, they are elongated. And because they're elongated, they like to kind of locally align, just due to the shape. So soon enough, you will see quite a lot of them moving together in one direction along the interface. And also at an angle which is not trivial, right? Not as low as individual cells. And so collectively, they assume a more non-trivial direction.
Sorry, I was writing.
Yeah.
I didn't. What does the red mean?
Just to see how differentially they're moving. Kind of post-processing for tracking.
But it seems that they are moving in the same direction.
Yes.
Oh.
Right. But now what happens is you will see the emergence of the fluid flow. So this is adding up all those individual disturbance fluid flows. You get this coherent fluid flow. And what you notice here is that the bacteria are moving in one direction, but the fluid flow is in the opposite direction. And this just happens because the cells are kind of aligning due to the shape, and then they push fluid flow back with their flagella in the opposite direction. So this fluid flow is what the flagella are generated collectively. And this still starts to make sense, right, in the dilute kind of drop. But what happens when you have a drop full of bacteria, right? And it was a little bit controversial when it came out because a lot of people said, well, this is very dense. Any fluid dynamics is very screened out, right? It shouldn't have as big of an effect. Right? And but it's still there, so it takes or longer in the simulations than in experiments to organize, but eventually you start to see some order, starts at the boundary. So same as with the dilute drop, you start to see packs of them moving together, and the lining, right, due to the elongated shape. Soon enough, you see a lot of these layers. This is 2D simulation, so there's layers. They're moving, and then the rest falls into shape really quickly. Now, one thing you will notice is that I've colored I put kind of a head, a black dot to indicate the direction of propulsion. But if I hadn't put those here, and I wouldn't put this fluid flow, all you would see is the bodies of the swimmers, right, moving backwards in the bulk, but the ones near the boundary moving forward. So, and that's what you see in the experiments, because you cannot tell the individual flagella, you cannot see them. All you see is the cell bodies, right, being moving here, and some of them, right, you cannot tell the direction of propulsion. So this is what we see, right, in the simulation, and I go to the experimentalists, I said, this is what I see. And they're like, no, go and do this again, this can't be right. I tried many times. Their initial interpretation, right, is that, well, the cells in the middle are moving backwards, so they must be pointing that way, right, swimming that way. And that was saying something different, saying they're all pointing outwards, but the fluid flow is pushing the ones in the middle backwards. The ones in the middle are swimming against the flow. The flow is stronger because there is more cells, like on the outer surface. And dynamics obviously was making this happen. They didn't believe me, so they checked with new experiments. So they got this bacteria. Yes, they got them from Howard Berg's lab, which is male. So you can color the cell body and the flagella and image them at different times, and then can figure out where they are in comparison with the others. So figuring out not just the direction of motion, but also the movement overall. Right? So the way they're pointing and the way they're moving. And so that the cells at the boundary are moving in the direction they're pointing. But the ones in the bulk are moving backwards. Okay? And since like all the other factors are ruled out, that has to be due to hydrodynamics. Okay? So it's like proof by experiments. Hydrodynamics matters. Now with simulations, one can do things like turn on and off different factors to figure out which one is affecting things. So if you turn off the hydrodynamics completely, even if you have ellipsoid swimmers, like elongated swimmers, in some cases you might be able to get them moving, but you definitely don't have the boundary layer. That's just due to hydrodynamics. If you make them circles or spheres, this is like squirmers, even if there is hydrodynamics, you don't get any circulation because circles don't align, right? They have trouble aligning. So you need both, right? The elongated shape and the hydrodynamic interactions. And different communities took different messages out of them, right? But one needs both, right? Not just the elongated shape. So where do you see this organization? The drop size has to be in a certain range. If the drop is too small, they don't have enough space to turn around and organize. And if the drop is too big, then it kind of starts to revert back to this quasi-turbulent state. So the drop has to be in a particular range. In this case, the drops with Bacillus acilis had to be 30 to 70 micron. Right? So these are 5 micron long swimmers. And this shows the length scale, right, for regulating the motion by using confinement, at least with the drops. So next we put them in racetracks. So this circular racetracks. And if you look closely, right, so of course you have bacteria moving in one direction. If you look closely, you can even see at the boundary some motion. It's just much more subtle. So they flow in one direction with quasi 50-50 chance, right, going left or right. So how do we try to figure this out? Well, locally, this is a channel, okay? But there's no impulse flow. For simulation, that's easy, make it a periodic channel. So cuts down a little bit on the cost. So what happens? Right? Start the disordered state, and soon enough they organize. You will see some cells moving together along the boundary, again at this non-trivial angle. And then they generate fluid flow, right, with their flagella in the opposite direction, and that's the fluid flow that it's carrying the ones in the middle. It's a weird form of collective motions, right? The ones in the boundary are generating fluid flow to move the friends in the middle. And is that the result of the shape? Both the shape and hydrodynamics. You turn off the hydrodynamics, you'll just get local aligning perhaps, but not this collective motion, right? This flowing. And again, check with experiments, and indeed, the bacteria at the edge, right, are moving one direction, but the ones in the bulk are swimming against the flow, right? Even the overall moving backwards. It's kind of again proof by experiments. So where do you see this flowing? Well, if you make the channels narrow enough, then you start to see kind of more or less uniform flowing, almost quasi-Poisson flow. When you start to increase the channel width, you start to see more fluctuations, and then at around 70 micron, this flow breaks down, no more flow, and we start to see the emergence of these vortices. They really like to go in circles, right, in vortices. And beyond that is turbulent. But again, you see this 70 micron. So that's the length scale, right, appears again. This is saying the same thing, right? So if the channel for this particular swimmer is under 70 micron, you can get this flow. Right? And I was very curious about this 70 micron. I get asked a lot by the continued theory people, like, what about the length scale? Well, it's about this swimmer, right? Only. Can't talk about all swimmers in general. But curiously, this was the same length scale of the transient vortices that you see in kind of unconfined turbulent suspension. So I hope someone will check this with another species of swimmer and kind of see the unconfined turbulence, see the size of the vortices there, and try to confine swimmers, right, under that size and see. I suspect, right, I mean, here that length scale is just, in simulations, it's just emerging due to the, just due to a few parameters, right? So it's a balance of those. So I keep saying this like a note of bacterial confinement. I mentioned this, like we see this, right? The movement, the vertical or channel movement, but that's only for Bacillus subtilis for these geometries. Wilson Poon had a paper with bacteria in a sphere. He's like, I don't see a vortex. And well, why was that? First, he used E. coli, which are Lampro bacteria. Cell body is maybe aspect ratio 2. And as you know, the closer you are to spheres, the tougher time you have a lining. And 2, it's a sphere. The sphere does not have an obvious axis of symmetry, unlike this cylindrical drop, which that is defined. So I'm waiting for someone to kind of try these experiments with different type of bacteria and check this systematically. Different shape bacteria. But getting back to the numerical methods, the method is versatile. Right? Within reason, it can be modified to look at a few different things. For example, if you're an engineer, you know about peristaltic pumps. Right? So you have a material moving down a pump, right, that is being pushed through. So if we put swimmers, right, and then we also have this. If we have these theoretical movers, here we have pushers and pullers, not only are they being transported by fluid flow of the pump, they're also collectively moving and interacting with each other and the pump. So it's a complicated fluid interaction problem, but we can simulate this, right? So moving boundary. And interestingly, just with simulations, one can see, right, that in pushers you get kind of things mixed. In puller suspensions, you don't get as much mixing, they just pull some fluid with them as they move. And in these movers, so they're kind of ghost swimmers, they self-propel but don't affect the fluid flow. They're just being moved along, right? And this is the dye field that they can't really mix. So it would be interesting to check with experiments, but this gives some idea. You can use swimmers to do some work. Right? Maybe mix some material or move something, right? Enhance, transport, or whichever is the application. Right? And so there is like that potential. On the other hand, complex confinement, numerically that's tough, right? If you want to handle the hydrodynamics at the boundary, not just the interactions, and do that with some fidelity, and the geometric confinement being something non-trivial, it gets more complicated numerically. There's always a cost and benefit, right? So how detailed can you have them? And so we have work kind of going on. We can use this inverse boundary method in complex confinement. Now, why? It's because the experiments are in complex confinement. So if you want to study those, you have to build the numerical tools that are appropriate. Last but not least, we can even have some scalar field, right, that is under this. And I use this example. For example, if you have bacteria, one thing they do is chemotaxis. So each of them produces a signal, and the other sense that and move up the gradient. So this is not quite bacteria, because bacteria do run and tumble for chemotaxis. So this would be more kind of chemotactic of photophoretic swimmers. So here is just neutral swimmers, so these ghost swimmers that don't disturb the fluid. They're each kind of releasing chemical, and eventually you see they aggregate. Chemotaxis mode for the way to aggregate. So they aggregate into the spots that are quasi-circular and pretty much static. What happens if you include this fluid flow? Make them pushers. Okay. Now, not only they are releasing this chemical, but they also disturb the fluid flow. This fluid flow is going to move other swimmers, but also this chemical they're producing. So you get aggregation, but you no longer get this circular static spots. You get dynamic aggregates. Dynamics. I mean, I picked the parameters for dramatic effect, but to show that you can get various dynamics, right? Couple dynamics of all of these processes. Any modeler or simulation person should show you the good things about their method and the bad. So this method, the inverse boundary method, is good. You can simulate thousands of swimmers. You can vary aspect ratios. You can have mixtures. You can resolve collisions in various shapes. And the dynamics is coupled with the fluid. The other can do 2D or 3D, and can do geometries. Right? And most importantly, it's versatile. So if you want to try non-zero Reynolds number, maybe not too big, and viscoelastic flows, for example, which in experiments is the case, one can do that. But it can get computationally intensive, depending on how much fidelity you want, like on a swimmer, and also the boundary. The fluid solver is on the grid. Confinement, right? You can deal with various, that's another method. But the way this model I presented, we're only modeling this far field fluid disturbance flow, right? This dipolar fluid flow, which you get by just two opposing Stokes laws. Now, depending on the application, maybe you're more interested in the close contacts, or you want to resolve more. Well, then you have to refine the swimmer, right? Add more points per swimmer. For example, like the raspberry method, so you kind of include more blobs, not tracing just one. But then it gets computationally intensive because you have to keep those blobs together, and you're tracing, right, more points per swimmer. So they all have pros and cons depending on what one can do. I mentioned that we also do models, right? So at the same time, we're finding models, like which models can be good and used for different reasons. And one model that I really like a lot is the bead and spring one. It's been around for quite a bit, by the group of Michael Graham, of course, inspired by dumbbells. So you have two spheres connected by a spring, and in one of them there is a flagellum force. So if it is a bacterium, that would be the back bead. If it is an algae, I guess that would be the front bead. By doing balance of forces and torques, right, you can get the equations of motion. And now for each swimmer, we're only tracing 2 points, just 2 beads. So not too many points per swimmer. So this is what this is saying. And then there is hydrodynamic interactions, which here are included with Stokes' lead. And there is a spring, so it gives it a little bit of flexibility, not stiff. So 2 equations per swimmer. And you can have collision forces and spring. One thing though that model did not include, but becomes important, is that bacteria rotate their body and counter-rotate their tail. And so they have this counter-rotation. So you have a torque dipole in addition to the force dipole. So we can add torques to the head and tail beads. And so if you make the beads equal, well, that's kind of rudimentary bacterium. But if you want to try a rudimentary spermatozoa, you can make one bead a little bit bigger to kind of approximate this asymmetry of the spermatozoa. So it's still very, very basic model. And the fluid flow, if you cut across the 2 beads, right, is this force dipole. But if you cut across these beads, you will see the fluid flow right due to the rotation, counter rotation. Right, so the force dipole is the dominant one. And you'll say, well, does this matter? Well, here we have 2 swimmers that are moving together, and the top ones don't have a torque dipole, so they move together and go swim. But if you have a torque dipole, they don't go swim. They kind of roll over one another and then get out of plane. And it's difficult to do with the hand, but it's this one goes up and this one down. Right? So their co-swimming, as you would see it in the lab frame, is just short-lived, not prolonged. And I mentioned this is the basis of a lot of continuum theories. But this becomes extremely important when you deal with surfaces. Why? If you don't have this torque dipole, the swimmer hits the surface and just moves straight. But with the torque dipole, it starts to rotate on the surface. And this is what bacteria do, and spermatozoa. And so you have a clockwise rotation at the no-slip wall. Right? And no stress interface is the other direction. Right? And there's been some work like looking at this circling, also the angle, right? It's not flat. So it's essentially the model is capturing this basic fact. Remember, it's just 2 beads, right? But you still have the hydrodynamic interaction, right, with the images, right, from the wall. Right? So you have the green functions for the Stokes flow, the Stokes lead, and then the rod lead for the rotation, and then their images. So first thing we try, right? Straight wall is straightforward, right? But what about a sphere? So you have an obstacle, fixed in the piece. A swimmer hits it, right? If it has a torque dipole, the deflection is no longer in that plane. So you can deflect the movement. You start to make this bigger, this obstacle. You see the deflection. The swimmer swims alongside the surface and then scatters away. And then, interestingly enough, once it hits a critical size, it's trapped. Okay? So once this obstacle is big enough, the swimmer just is trapped here and swims alongside. Here along an equator, here tracing a smaller circle. And one can even play these games of, well, we have the swimmer inside the solid cage. You would see the same thing. And the method of images for a sphere, for Stokes lead, right? The sphere, it's a various paper, just the formula alone, it's more than half a page. So it starts to add like a lot of computational complexity, right? You're tracing 2 Stokes lead plus 2 rod lead, their images. So if you want to do a realistic problem, right? swimmers in a porous media. For example, these gel beads, right? You have bacteria moving in this, among the spherical beads. You don't really want to do simulations like that, right? That resolve because it's just spheres, it's complicated. But these were kind of inspiration, right? There's been a lot more work done in kind of confinement, whether that is this porous like structure, where you can see that the bacteria kind of hop from chamber to chamber, right? So it's more like they're trapped in a chamber, and then they see an exit, hop to the next chamber. There is work done on swimming among ordered pillars around them. And also, even if you have very dilute colloidal suspension, it can also affect the individual and collective motion of swimmers. So essentially, kind of confinement, roughly defined, is important. Another thing I mentioned is that you can adapt this to make a rudimentary spermatozoa. And this is because my colleague in Warwick, Vasily Kanzler, he does a lot of work with spermatozoa, so started to look into this, and we talked to experimentalists. Right? In the lab, we just have pillars and channels. So it's a rudimentary spermatozoa, right? If you send it along like a straight wall, right, to do the circling, but we can do pillars, right? So we can also follow this. And he will say, oh, wait a minute, what is the method of images, right, for a stock sled cylinder? There isn't one, right? There is no closed formula. So at this point, we already get into approximating the image system. So, but that, making a reasonable assumption what is a good fidelity, one can still use this model to learn things. So if you have a pillar that is very, very thin, the swimmer goes there, barely acknowledges it, right, but then continues on its way. If the pillar is a little bit larger, the swimmer can go there, swim alongside it for a little bit, and then scatter away, depending on how big this is. As you'd imagine, if the pillar is big enough, right, there will be a critical length scale where the swimmer is trapped. In here, I haven't added round motion, but you can think about what are length scales, right, to choose if you want to entrap swimmers. Right? For the previous one, people have used kind of pillar forest to deflect the bacterial colony at a particular angle. So these are important for applications. I'll mention an interesting application of this. So here you have two surfaces. And you can see that the swimmer is swimming kind of at this intersection of these two surfaces. Right? It's attraction to both surfaces. Now, maybe you know, maybe you don't know, but there are bacteria that hunt other bacteria. And complicated name. And you think, right, you are a hunter trying to chase a prey, chasing in 3D, kind of tough, right? Even chasing in 2D, not necessarily simple. So where do these predator bacteria find their prey? Exactly here, right? So if you think of obstacles, the chances are high, both for the predator and the prey bacteria, that they will be like where these two surfaces meet. So that's where they actually find their prey, because then it becomes a 1D search problem, right? Just kind of searching alongside that. And so it's an interesting thing that one can see, right? Anyway, we are looking at kind of building these methods for collective motion, right? So this is collective motion of spermatozoa. You get this quasi-turbulent behavior for these as well. And remember, these are very different geometrically than bacteria. Bacteria have a solid body, and the flagella, spermatozoa have this undulating flagellum. And just like what you saw with the channel, with the bacteria, if you put spermatozoa in a channel, they will flow, right, overall, with the width of this channel. But the mechanism is the same. You will even see here, right, the spermatozoa moving in one direction near the wall, and is getting carried back by the fluid flow. So it's still a hydrodynamics effect that is doing this. So we are building more methods for this, again, with more confinement and complex confinement. And I mentioned chlamydomonas, so I'll talk more about this. So chlamydomonas was the other type of swimmer, the one that did the breaststroke, pulling its body forth. And you can model it with 2 beads, or just with the ellipsoid. That's great. But it was noticed in experiments, again by the Goldstein group, that the fluid flow for this, and this is average fluid flow over the stroke, you can approximate it by 3 stroke sleds, not 2. So you can put 1 stroke sled at the body and 1 each with half the strength near the tip of the flagella. And this captures the fluid flow better. So this gives an idea. We are dealing with beads and springs, right? Instead of having, the head and tail beads, you have the body bead and like 2 arm beads, so 3 beads, right? In this kind of triangular configuration, and all connected by springs. And it's also computationally difficult because you're just tracing 3 beads, right? And then hydrodynamic interactions. So the fluid flow looks right, it's fuller one. And what happens? Let's see if we can play this. If you send this swimmer right near a wall, it hits the wall, the arm that touches the wall can compress a bit, and then it will scatter away. You will see that the angle of scattering is very different from the angle of incidence. This has mostly to do with the geometric shape, right? This kind of triangular shape. So it kind of hits this, takes some time to orient, and once it's free, then it will scatter away. And algae also do something like this. I mean, there is repeating as well of the flagella, kind of touches the wall and kind of swims alongside it, reorients and moves away. So this roughly captures it without the flagellar bit, just by using hydrodynamics and this triangular shape. And now, what if you send this algae in a too steep of an angle? It gets stuck with the flagella, well, at least in the simulations. And if the incident angle is too low, kind of barely acknowledges the wall, but it will be a trajectory, but may not actually touch it. And then in between these two, there's this scatter regime, right, that we showed, where the swimmer goes there, and the outside angle is it scatters away after some time at the surface. So we thought, okay, let's measure the resident time, like how long it spends at the wall. And so if it's not touching the wall at all, that's 0. If it's scattering, you're going to have this quasi-linear dependence, right, on the incident angle, because it takes time to turn. But then, if it's hitting the wall, kind of with the flagella, and it's stuck, well, it's infinite time. And one can add some Brownian noise to the beads. I mean, if a lot, then maybe it can scatter, but you always have this trapping here. And then you look at the experiments, and you see this quasi-linear dependence, but then it's finite, right? They don't get trapped. So the residence time or impact time, it's finite for algae in the experiment. So why is that the case? Well, algae do something funny, so they also rotate slightly. And we didn't have this rotation in, but if you hit the wall and you're trapped, right, just by rotating, the algae reconfigures itself, so it's able to escape. So the same with this other simulation. So if it is trapped, turn on this spinning, that enables it to configure itself in a more favorable direction, so then it can scatter away. So this is an important detail, right, if you want to study the interaction with the wall. Right? You have to take the spinning into account, the triangular geometry, not just the hydrodynamics. Right? You can't do the spherical cow solution and capture this detail. Right? So you want to study the algae properly, right? These are important. With the bacteria, it was the elongated shape, right? Counter rotation, in addition to the hydrodynamics. Oops, this is playing. And we saw this with experiments using a cylinder and a popsicle. So here you see the algae, and it's approaching cylinder, and you kind of see the arms, but you can't quite see, I guess, the rotation. But it swims alongside the cylinder. You see from above for a little bit, and then it will scatter away. So you have these three distinct regimes, right? The one where it is trapped and then scatters by spinning, which you see in simulation. The one where it's touching and scattering. And then there's this physicist gave the name hydrodynamic escape, where it kind of barely acknowledges the pillar, but it goes in its way. And if you think from a theory point of view, if you want to make a model without doing all these detailed simulations, right? This is a three distinct regime that you have to tell if you're making a minimal model, like how to interact with the boundary. Right? It's dependent on the incident angle. Depending how it approaches a boundary, it has three different outcomes. Right? This is tough, especially for, say, if you're doing continuum theories, right? The interaction of the boundary. And last I mentioned the phoretic rod swimmers. So these are usually rods or spheres, and usually half-half. So gold and platinum, a few microns long, and so usually ganospheres. And we wanted to kind of look into this, especially the effects of hydrodynamics. Now, if you put this in hydrogen peroxide, they swim. If you think, okay, if I want to make a model of this, this is complicated. Obviously, you have hydrodynamics. They also are at the bottom of the chamber. Right? You have confinement effect, but there's chemistry, right? Because you have an exchange of ions, right, to make this propulsion possible, and doing all of that, like in a good simulation, it's tough, right? Even for one swimmer. So, how do you kind of do a model that captures something of this, right, without being too complicated? Well, the idea was that there is an exchange of ions here. So if you think in terms of fluid dynamics, there is a slip layer, right, somewhere at the junction of the two metals. Now we don't know how big this layer is, right? So for simulation purposes, just assume half. So without solving for the chemistry and the all of that, we reduce this to a hydrodynamics problem, right? So we have swimmer with a fluidic layer, right? So the propulsion works because of that. And now by varying the portions of the gold and the platinum, you can move the position of this slip layer. So here, the long gold, the junction is in the front, the slip layer is in the front. Think slip layer is the actuation mechanism. So what's actuated in the front? It's the puller, right? And indeed, the fluid flow that you get for this is the puller fluid flow, right? Where the fluid flow comes in the front and back and goes out at the side. So we made the puller kind of swimmer for this. And if you do short gold, right? So the actuation is going to be slip layer in the back. What has the actuation in the back? Bacteria, pushers. So this is a pusher. Okay? And the fluid flow indeed indicates that. Fluid flow here, the streamlines coming in the middle, go out in the front and back. So essentially we can get pushers and pullers and then see the effect of this. Simulation-wise, it becomes a little bit more difficult because you have to keep this together, right? All of these blobs. And you want to resolve this a little bit more detail if you want to capture things. The question becomes a bit, the question that is of interest was rheotaxis. A lot of people that have studied microswimmers know that that's a thing, right, for swimmers, in fish as well. So they swim against the flow at the surface. And so here you have fluid flow, and the swimmers near the surface are going to go against the flow. Okay. And the person who did the experiment is Quentin Brosseau, who also was a postdoc here with me. And then for this work was at NYU. So just to kind of check if you don't have any, there's no hydrogen peroxide, any rods, right? They are immotile, so the flow just carry them, right? In the direction to move them. If there is no flow, they can go back and forth at equal probability. That's this case. But if you have both the flow and they are motile, they will stream upstream. And then the question is, well, which one of these types of swimmer is the most efficient, right, at geometries? Right? So these are micro robots. People have big hopes for them to be used in medical applications. Suppose you want to send them for drug delivery to reach a target, right, to go against the blood flow. Which type would you design? And it's not intuitive at all, but it's pullers, because they swim at a higher angle, and because it's a higher angle, they're more susceptible to the fluid flow. For geotaxis, you need the fluid flow to orient the swimmers. So pullers win for this. What's next? Gravitaxis. So for the original swimmers, right, the gold and platinum, they're very close in density. So so you want to kind of make a more substantial density mismatch. So rhodium in this case is more suitable. And if you build a swimmer like this with gold and rhodium, there is a mismatch from the center of hydrodynamics and the center of mass. So that tilts the swimmers. Right, so they're kind of, think about them as kind of bottom heavy. And then, but they're still trying to move, but in this case, they will move against gravity. So you can send them on an inclined plane, and they will go. On average, they will climb up. You can send the army of micro robots to move against gravity. So I mentioned this, I'm also going a little bit about the time, but we have ongoing simulations doing tens of thousands of swimmers. These are bead and spring swimmers in a periodic domain. In this case, pushers. I mean, the aspect ratio very, very small. That's why those really large scale structures. We can also do elongated kind of spiral cylinders above the surface. And it's computationally heavy because not only is there collisions that are resolved, but hydrodynamic interactions and with the surface. So this is periodic on the sides and just some artificial limit above, so they don't fly, they don't go away. But you will see that soon you will have these collective structures develop. A lot more of them are near the surface, just like that. And once there is enough of them, they will collectively swim. You have these flumes, bacteria. But this is just an example to show what simulations can do. It does take a couple of days to run in the flat irons of the computer, but it's possible. So in the meantime, one can modify these simulations and see. So what do I want you to remember from this? Interactions with the wall are complicated. You have an interplay of direct contact, which depends on shape, hydrodynamics, and any swimmer particulars, spinning of the head or the entire cell. And wall interactions and pair interactions, you cannot describe them by a simple rule. Right? If you want to make a continuum theory based on this, it's complicated. Interactions with confinement reveal specific length scale. We get asked about this a lot from theory people. Well, they show up, right? It's they're there, right? And those length scales become important if you need to see applications. So we try to extend these existing models of bacteria to look at various confinements and other things, hoping to include right physics, keep things versatile, and most importantly, tailor those to the swimmers want to study. But then understanding those, one can learn how to control them and the collective motion. So I will stop with this, and thank you. I went a little bit about.
Lots of systems covered. It's very interesting. I really enjoy it. We only have time for maybe a couple of quick questions. Yes, Matthias.
Excellent talk. On your Janus particles, the way you modeled them, I didn't fully understand. Like, it seems like you had like the two on the left and the right to me look very. No, sorry, the two on the right they look very similar.
So the sleep layer,
Yeah, the slip.
it's in different position.
Yeah.
This is the red blobs.
Right.
So those have a prescribed.
Now the two on the right though, like you move the slip layer, but like, do you keep the velocity constant? Or like
The velocity emerges by itself. Like it's related to the length of this sleep layer.
But they look like the same, right? It's just flipped. So
yeah.
how is that different?
The direction, right? So here the streamlines are coming off right from the middle, and here they're going.
But then in both cases you get movement to the right.
So here it's this, right, the sleep layer, you see the fluid flow, and here it's essentially where those streamlines are coming to the bodies.
I guess my question is on the right one, why doesn't it move to the left? Why doesn't the Janus
because there is a direction on this. When I mean sleep layer, there is a direction. I think of each one of these blobs, right, having a direction for the fluid flow backwards.
Oh, okay.
So
okay. So
The right one has like two eddies behind. You see the tail thing?
yeah. I thought slip you just mean like it's not a no slip.
It's a sleep layer, so it's not zero fluid flow, but essentially it has a constant fluid flow.
You enforce a direction for the ICS.
Yes. So in this, the propulsion emerges by
Mhm.
this. So this is essentially a squirmer
I see,
kind
I
of
see.
model.
Okay. Yeah, that makes
The
sense.
geometry. So, yeah.
Yes.
I think in the last few slides you mentioned periodic boundaries. Sorry, what is this?
Yes. So the periodic boundaries were on this side, right? So on the two lateral directions, right? It's if the swimmer is coming out here, then it will come back here. So it's just kind of the same copies of the same thing happening, kind of domain. And because it's difficult to kind of simulate free space, one, and two, a lot of these numerical methods, they are optimized for this particular periodic boundary conditions. So, FFTs.
So are you simulate them in a long channel? Is this the situation you try to simulate?